In this lecture, just we can understand this structure which is mentioned here, right? So this is the mesosomes where like tubule, vesicles, and lamella is there. It has been there, and cytoplasm is equal to semifluid, that we call that because this is the half fluid is present in this, and chromatophores where which is the chromato means what color, and phore means pigments, right? Which we have to phore means like as you can see that cell wall which is the rigid which is absent in mycoplasma, and cell membrane which is the flexible here, right? And this is the flagella, this is the flagella, right? There is also included bodies which help in storage, and here's a plasmid. We can see that extra chromosomal DNA which is double circular DNA is there, right? Which is a small here, and polyamines proteins and ST plus which stabilize the DNA, binds to DNA, and they are not the histone proteins, we have to remember. Ribosomes is present which is 70s here, which is bonded by cell membrane, not right? And nucleoid is also known as gonopore, not gonopore, genopore is mentioned, which is a single chromosomes of bacteria which help, which is the main DNA which is the negative here, which is also double circular, double standard circular DNA which is naked here. Okay, next, see, if we talk about the surface structure here, so flagella, how will we identify this? If bacteria is a motile, what is motile? Then it must have flagella, we have to remember. And if bacteria is not motile, that means there's no need of flagella, right? Okay, so in a prokaryotic flagella is different as compared to eukaryotic flagella. In a eukaryotic flagella, which is form of the tubulin protein, which is a complex protein, and if you talk about the prokaryotic flagella, which is form of flagellin protein, where the lacuna protein is there, that means it has been decreased. And flagella is a thin filamentous which is hair-like structure. Flagella is the extension of the cell membrane, okay, which is the flexible, and the flagella extend from cell wall. Parts of flagella, we can see that the first one filament, which is the longest part. Second one is a hook, which is a connect with this basal body and filament. Third is the basal body, which is at base. Next. Okay, here the villi is there, which is elongated tubular structure made of a special protein that we call that pleen proteins. Okay, yeah. So in a note, after conjugation, both bacteria become F plus here. Okay, which help. This is the pili, okay, and this is the F plus. Before it was something like that. It helps in the transport of the plasmid from bacteria to other bacteria. It is called conjugation here. Okay, and femora is a small, which is a blister-like structure, right, which provide the encouragement, right? That means grip. We can see that ability to bacteria here. It helps in bacteria to encroach to store the host tissue here, okay, and form the fibrin proteins here. Villi and fibrin have no role in motility, we have to remember. Okay, yes. If we talk about the inclusion bodies here, so it is a membrane less, you have to remember. It help in the storage. Inclusion bodies help in storage of different materials, where the phosphate granule, that means store the phosphate here, glycogen granule which is store the glycogen, cyanophasic granules which is store the pigments here. And if we talk about the gas vacuole here, so not a true vacuole but which membrane is there, right? The gas bubble which provide the buoyancy to bacteria. An example of gas vacuole which is mentioned blue-green bacteria and purple-green bacteria. Next, ribosomes, right? It is also known as a universal. What's a universal organelle, which is also membraneless organelle, and it is also called that pellet particles. Okay, at the time of 1953, George Pellet name was given. Is the name of scientist who discovered his teeny dense granules in cell. Electron microscopy needed on that time. Okay, and proteins factory, that we also call that to the ribosomes here because it makes protein from mRNA, that means messenger RNA here. Okay, we can see that mRNA and proteins, and when many ribosomes sits on a single mRNA stand, that means to make a proteins, then it is called polysomes or polyribosomes, we have to remember. This is the smallest organelle, which is 15 to 20 nanometer in prokaryotics. Okay. Next, as we can see that the universal organelle ribosomes is found in both prokaryotic cell and eukaryotic cell. So in a prokaryotic cell, which is 70s present, that means large is 50s and small is 30s. Okay, that could be converted here. The eukaryotic cell, the small is what, sir? 3, 4, 40, 50, and 660. That's all. Now, just we can remember the NCRT here. Bacteria cell may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella here. Bacteria show a range number and arrangement of flagella. Bacteria flagellum is composed of 3 parts: filament, hook, and basal body. The filament is the longest portion which extends from the cell surface to the outside here. Besides flagella, pili and membrane are also surface structure of the bacteria but do not play role in motility here. Do not play a role in motility here. Okay, we have to remember pili and fimbriae are also surface structure of the bacteria but do not play in role in motility. Okay, the pili are elongated. The pili are elongated tubular structure made of a special protein here. The fimbriae is the small. The fimbriae is the small and blister-like. Okay, fimbriae is the small and blister-like fibers sprouting out of the cell here. So in some bacteria, they are known to help to attach the bacteria to rocks in streams and also to the host tissue, as we can see that here. This is the extra point here. Monotrichos means here like lopotrichus here, then there are lots of there, right? And amphitrichus, we can opposite one. Here the amphitrichus and feritrichus here. Question, we can see the bacteria show a range in number of the arrangement of the flagella. Bacteria flagellum is composed of 3 parts here. Option 4. Filament, hook, basal body. Right. And just we can see that here, that which of the following is the incorrect for ribosomes. That means bad point. They have no real protein synthetics. That is wrong. Okay. Correct the sequence of the layers of the bacterial cell envelope. This is raised, but the correct answer obviously, or maybe option number 2, but we have to move forward here. Inclusion bodies, which is reserve material in prokaryotic cell, are stored in the cytoplasm in the form of inclusion bodies here. These are not bonded by the any membrane system, which is lie freely in the cytoplasm. Example, phosphate granules, cyanophycin granules, glycogen granules, gas vacuoles are found in blue, green, and purple green photosynthetic bacteria. Okay, we can see that question, a largest subunit of prokaryotic ribosomes 50s. Yes, gas vacuole are found in where? Blue, green photosynthetic bacteria and purple and green photosynthetic bacteria. Option number 3 is the correct answer. The longest portion of the bacterial flagellum that extend from the cell surface to the outside is called as filament. Yes. Here we can see that we can read NCERT here that ribosomes are inclusion bodies here. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size, and made up of 2 subunits, 50s and 30s subunits, which when we represent like present together form 70s prokaryotes. Ribosomes are the site of the protein synthetics. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes or polysomes translate the mRNA into proteins. Yes. The question is asking from the statement given below, choose the correct option here. So we can see that here the eukaryotic ribosomes are 80s and prokaryotic ribosomes are 70s. Yes. Each ribosome has 2 subunits. Yes. 2 subunits of 80s ribosomes and 60s and 40s, while that of the 70s and 50s and 30s. Yes. But here we can see that it's not 70 years, not 30 years. That means from D and E is wrong. A, B, C is the correct answer. Next, ribosomes. Here we can see that which is the granules, granular structure first observed under the electron microscope as dense particle by Gilbert Perret. They are composed of ribonucleic acid, proteins, and not surrounded by any membrane here. Eukaryotic ribosomes are 80 years, while the prokaryotic ribosomes are 70 years. Each ribosome has 2 subunits, larger and smaller subunit. Here we can see them. 80 years ribosomes and 60 years, 40 years, while that of 70 years ribosomes are 50 years, 30 years. Here S is a similar Sternberg's constant, that means unit, stand for the sedimentation coefficient. It is indirectly measure in density and size here, right? Right. Ribosomes form of the ribosome plus proteins. Ribosome is an organelle which is form of backed proteins and form of proteins here, right? Yeah. Now we can see the NCERT here. Prokaryote ribosomes are 70 years, has 2 subunit, 40 years and 68. Wrong. Because within the cell membranes, ribosomes are found not only in the cytoplasm but also within the organelles, chloroplast. That's also wrong. Both statements are wrong here, right? Next, which type of the vacuoles provide the buoyancy to bacteria? We have read contractile vacuole, not here. The gas vacuole is the answer. Correct. He's asking here that mesosomes is formed by the extension of the plasma membrane into the cell. The pili are elongated tubular structure made of the protein. Flagellum is composed of filament and hook, basal body acid. Option number 1, 2, 3 is the correct answer. If you remove the fimbriae from the bacterial cell, which of the following would we expect to happen here? Obviously, the bacteria would not adhere to the host tissue. Wrong statement is asking for ribosomes here. Ribosomes was observed under the electron microscope by George Perret. Yes. They are composed of ribonucleic acid and proteins are surrounded by the membrane. No, that's wrong. So option number C, only 2 is wrong. Protein synthesis in animal cells occur here on ribosomes present in the cytoplasm as well as in the mitochondria here. Okay. Many ribosomes may associate with a single mRNA to form multiple copies of polypeptides simultaneously. Such a string of ribosomes are formed as polysomes, obviously. Select the wrong statement here. Obviously, cyanobacteria lacks flagellated cell. Yes. Mycoplasma is a wall-less microorganism. Obviously, bacteria cell wall is made of the peptidoglycan. Yes. But pili and fimbriae are mainly involved in protecting bacterial cell. No, that's wrong here. So that's all. Thanks.